presentation today, and delighted to have as our presenter Will Orndorff. And Will is the Karst Protection Coordinator for the Virginia Natural Heritage Program in the Department of Conservation and Recreation. And he holds a master's degree in geology from Virginia Tech with an emphasis in structural geology and tectonics. Let's see, his career has led him down an interdisciplinary path that includes hydrology, biology, geomorphology, and analysis of spatial data. There's lots more I could add about Will. All of our presenters today just have a really rich background, but Will, I'm gonna turn it over to you and let you get going.
Okay, thanks, Bev. So the only relevant thing that I would add to that is that I'm a native of the Shenandoah Valley and Shenandoah County, and I grew up on a farm just north of Strasburg, right near Cedar Creek, right next to one of those mines where they mined the Newmarket limestone Dan talked about. So I grew up drinking karst water from a well in the Newmarket formation, and you know, so I've got all the valley credentials there. With the name Orndorff kind of gives it away.
I wondered about that. That's a pretty prominent name here. All right.
Sure is. And I guess we're all related, but we don't act like it. So I don't know. Anyway, so I'm going to talk to you about cave life. And the reason that we have a state employee in Virginia who works on cave and karst issues is because of this cave life that we have. This outstanding biodiversity is why the Natural Heritage Program can justify including what we do in our mission. So even though I get dragged into a lot of other protection things, it's really kind of centered around cave life. So anyway, you can see an example here on the intro slide of a few of the interesting cave animals. They're not very big. That crayfish, we don't have that particular type of crayfish in Virginia. That's about 3 inches long. Everybody else here tends to be anywhere from 3 or 4 millimeters to about a centimeter and a half long, depending on the species. I intentionally did not put a scale bar in because they do have some size ranges, and it just kind of detracts from the view. One of the things I always like to point out is these things were really big, they'd be really scary. All right, let's see. All right, my slide is not advancing. Come on now, slide, you can do it. Hold on. All right, let's try it now. There we go. Okay, so when we're talking about things that live in caves, you know, people like to categorize things. And so we have these 3 terms based on the relationship of the animal to the cave. And we're not, when I say things that live in caves, I want to talk about animals today mainly. Troglodytes are cave guests. You find them in caves, but they're not limited to caves. They don't necessarily depend on caves. Some cave troglodytes include some certain salamander species. They include cave rats, like you see here. That's Allegheny wood rat or pack rat. They also include things like raccoons. So they're animals you might find in caves. They don't really depend on caves. Troglophiles, on the other hand, are animals that seem to really like caves. You see them in caves more often than other places. And the cave may well serve a critical role to that animal in its life cycle. Examples like that are this cave salamander that you see there on the right in that pretty little rimstone pool. Certain bat species depend on caves for part of their life cycle, cave crickets, things like that. But all of these animals have in common is that they go in and out of caves. So they're not limited to caves. I'm sure certain cavers consider themselves to be troglophiles and not just troglodytes, that they sort of, their life depends on getting in and out of that cave. So troglophiles, they need caves, but they spend time in and out. Troglodytes, they like caves. Troglobites, on the other hand, are cave-limited animals. These are animals that are only known from underground habitats, and they depend on that underground habitat for their existence. And so from my perspective, the most interesting, and the reason we have an interest in the Natural Heritage Program are these troglobites. Now we do study bats a fair amount, and that's a separate talk. I'm not going to talk to you much about bats in this talk because the troglobitic fauna is really where the action is. Okay. And so for our troglodytic fauna, we're mostly talking about invertebrates. Now here's a couple troglophilic invertebrates. These are some invertebrate species. You have an insect, a cricket up at the top. You have a spider at the bottom. And these invertebrates, you find them in caves. They seem to be dependent on the cave, but they can move in and out of the cave. They're not limited to caves. And a lot of the things that became troglobites, cave-limited animals, probably descended from troglophiles, animals that were in and out of caves. And over time, they became an advantage to be able to never have to leave the cave. So troglophiles are interesting in their own right. Okay. And you can see some examples here: cricket, spiders, daddy long legs, some types of flies, fungus gnats, some millipedes are troglophilic. So, it gives you an idea of what we're looking at with troglophiles. Troglobites, on the other hand, are really interesting. They all have adaptations that reflect specialization for a cave environment. One of the big specializations that they have is what were mistakenly determined as to be retrograde evolution or regressive evolution by some biologists back in the 19th century. For instance, they lose their sight. They lose pigment. People used to say, well, this is a negative thing. Well, actually, if you think about it, these are positive things for a cave environment. Why waste metabolic energy to make eyes if there's no light? Why waste metabolic energy to make pigment if there are no damaging ultraviolet rays, if it doesn't serve a purpose? I always have to point out here, this cave beetle over here, that's actually a pigmentless cave beetle. The color is due to a phenomenon called sclerositization, which is where the reason that pigment is there is it actually hardens up the exoskeleton, and the color is just incidental. But typically cave things are translucent or pigmentless. They're blind. They tend to be smaller than their surface ancestors. They also tend to have different reproductive strategies. For example, they'll lay more eggs, or excuse me, fewer eggs, and they'll do it infrequently. So the value of each offspring is a lot higher to a cave animal than it would be in most cases to its surface equivalent species or related surface species. So these cave invertebrates are really, really interesting in their own right. You can see some of the terrestrial or land-loving cave invertebrates include groups from millipedes, beetles, diplurans and springtails are both sort of primitive relatives of insects that still persist. Harvestmen, daddy longlegs, spiders, of course, we all know, and pseudoscorpions, which are actually one of the oldest terrestrial invertebrate lineages on the planet. All these things are commonly found in caves. In the aquatic realm, we have a little bit less diversity at that level. There's amphipods, isopods, copepods, all those are crustaceans. And then you got snails, which of course are gastropods. And these animals live in the aquatic and cave water habitats. All right, some general characteristics, like I mentioned before, for these troglobites is reduced pigment, eyes are reduced or absent, they're smaller, they may have enhanced development of other sensory apparatus. So if we, and we'll see later, but if you look at a lot of these troglobitic invertebrates, what you'll see is they have little cilia all over their bodies so that they can feel their way around. They also have elongated antennae. Their antennae on average are more than twice as long as their surface relatives. So they have other ways of finding their way around their environment since they can't see. And again, like I mentioned earlier, they have different reproductive strategies. The photo on the bottom there is a flatworm, or a planarian, as we like to say in Latin. But flatworms are great. You find them in cave pools. And if you touch them, they kind of suck up into what looks like a little ball of snot. You wouldn't even know it's alive until it's relaxed and crawling around. So those are kind of neat. That one's about an inch long. They can get pretty big. And by the way, for the gross out factor, every now and then somebody pulls one of those up in a well in the Shenandoah Valley. All right. So how do these things become cave animals? Now, there's different philosophical outlooks. You can say that, well, they were created that way, and that's fine. But the business of biologists is to find out how they evolved or were created or whatever, how they got to be that way. So what you can see here is a great example of progressive adaptation to the subterranean environment. In the top left, we have a species called Lyrceus hargeri. So Lursius is a genus of an isopod. It's a crustacean. And these things live in springs all over the upper Tennessee River Basin. Well, the most common ones that are widespread have well-developed eyes. That's what those black patches are. That's the eyes for the isopod. That's its head you're looking at. And it's facing upwards. Okay, so there's Lursius hargeri. Well-developed eyes, lots of ocelli or individual eye cells. Sort of your classic surface crustacean. As we go down, though, the next one, this is a new species from a cave called Hugh Young Cave in Tazewell County, Virginia. And you can see the eyes are about a fifth or a sixth of the size of the ones in hargeri. You come down to the next one, you see a new species from Singleton Cave in Washington County, which is down where Bristol is. You can barely make out where the eyes were on this guy. And then as the final one on the lower right, you can see the eyes are completely gone. So almost everything else about these animals looks very, very similar, except there's progressive loss of pigment and loss of eye function. Again, why waste metabolic energy on these adaptations if you don't need them for the subterranean environment? This is just a good place to see this progressive adaptation. And here's just a picture sort of showing you what some of the spectrum looks like. The cave adapted ones are in the middle, as I think, since everybody's muted, I'm not going to ask people to chime in. But I think you can all see that this is the cave adapted guy, and these are his various surface cousins known from the springs in the area. You can see they're really quite beautiful animals, and and quite a stunning array of diversity, which is why we're interested, and particularly in these cave adapted ones. All right, so what's the biodiversity significance of cave fauna? So biodiversity is the number and nature of animals that you find or organisms that you find in a place. The troglodytic cave fauna occupies a particular place in the world of biodiversity. So any particular cave you go to, if you were to say, let's look at all the species in this cave, and let's see how many we've got, we would come back with a pretty non-biodiverse place. We would find 10, 12, 15 species in a typical Appalachian cave. If you look 10, 8, 12, 15 trips in there, you looked around. So you're not going to see that many. So at the cave level, this biodiversity isn't so impressive. However, if we go to the state of Virginia level, all of a sudden it becomes really impressive. And that's because cave animals have a high degree of endemism, which means, known from only the place to which they're endemic. And these endemic areas for cave animals can be as small as a single part of a single cave. Since they're so endemic, you get a lot of species over a relatively small land area because you've got lots of caves that are each separated from one another. So one of the things that cave biologists do that really most biologists would find rather boring, but we find pretty interesting, is we make these species lists, which is like, here's what we found and here's where we found it. And the most recent one that came out was in 2013. Now, the guy that you see there on the right, I tell you, I grew up near Strasburg. This guy is about the same distance south of Newmarket. He grew up in Stanton. And this guy is John Holsinger, another good Shenandoah Valley name there. Well, John Holsinger turns out to be the father of Appalachian cave biology. He got interested in this stuff back when he was a student at Virginia Tech in the 50s, primarily in caves to begin with, and then he got interested in cave biology and just kind of took it from there. So he, near the end of his career, published this second comprehensive list of the cave fauna that lives in Virginia. And in that book, had 121 different terrestrial cave limited species and 47 all known from Virginia, and 90% of them known only from Virginia. So it's a really special thing to have this many. I think in the United States, we're tied for second in terms of the most cave limited species of any state. So, we rank pretty high there, which is kind of fun. And part of that's not because we have so much karst, but we have such diversity of caves and karst in that area that Dan showed you earlier. And again, these between the terrestrial and the aquatic species, we find more terrestrial species because the terrestrial habitat tends to be more isolated than the aquatic habitat, which can carry animals from one cave to another. in situations that would drown. So here's another picture of Dr. Holsinger. And one of the reasons he pushed to have caves protected by Virginia state law was this extreme endemism and significance of the Appalachian karst. So we had 168 species in 2013. Since then, we've discovered another 25 or 30. And a lot of these are currently having their formal descriptions prepared. So, if you want to go somewhere and kind of cheat and be able to find new species, go into cave biology. You will find new species. All you've got to do is spend a few days looking, and you're going to have a new species in your vial. And if you're lucky, you'll find somebody to describe it. And if you're persistent enough, maybe you'll learn to do the taxonomic work on your own. But it's a real treasure trove of rare species. So let's kind of go through here and see some pictures and look at some of these groups. The cave beetles, just like in the surface, beetles are an incredibly diverse group. And these carabid beetles in particular are diverse. And what we see over here is a pseudoscorpion, which is the common genus of carabid beetle we find in Virginia caves. We have 39 described species. Another 12 are being described right now. And I've got another, I don't know, dozen or so that are, well, this is something different, but we don't have the money to describe it right now. So by the time we get done, if you were to project, there's probably over 100 different species between Virginia and West Virginia of cave beetle. Millipedes are just slightly less diverse. We got 22 known from Virginia and 9 known from West Virginia. Again, that's just the described species. There's several in the queue waiting to be described. So again, these really species rich at the level of the region. And what I like best about this talk are these cool pictures. This is a close-up of a cave beetle. This guy is called Pseudoscorpion parvicolus, the thin-neck cave beetle. And he's known from only one cave in Virginia, in Frederick County, a cave called Ogden's Cave. And he was thought to be extinct. He was known from Hupp's Hill Caverns, but all the development, everything that took place there, it seems like degraded his habitat. And when I picked this beetle up in Ogden's Cave about 15 years ago, I suspected this might be it. And when it came back that this was indeed what we'd found, we were very happy. And because of its rarity, we were able to justify spending money to develop a natural area preserve there at that site. So that's a pretty cool beetle. It's done some good, in my opinion. Here's a millipede. This is a cave-adapted millipede of the genus Pseudotremia. Not to bore you with Latin names, but there's a few different genre of millipede, cave-adapted millipede. And they're just really awesome animals. And they're scavengers. You can see, if you look closely here, the cilia, the whole body is just coated with these little hairs, which is what helps it find its way around. These guys are the real terrors. These are the ones you could, if you were like H.H. Geiger, you'd make like a horror painting of this thing, human size. This is a pseudoscorpion. And I don't have a picture with the claws extended, but if you extend the claws for this guy, they come out to be longer than the body. So if you were to encounter a human-sized pseudoscorpion and walk up to, be talking to it up here near its pinchers, and you were all focused on not getting pinched by the jaws, it could still ambush you from behind. And that's how these guys work. They're ambush predators in caves. They sit around, they hardly move, and they have those claws out. And when things get in the little corral between the mouth and the claws, they don't leave. So again, really neat group. You can see here again the sensory apparatus on it, the lack of pigment. Just a fascinating animal. And we're really happy they're not any bigger than they are. There's another one, another pseudoscorpion. Again, the cilia over here, really characteristic of cave-adapted animals. Okay, this guy, now this is the Shenandoah Valley endemic, and this is undescribed. This is what's called a pigeon diplurin. So if you look up here, it's got 6 legs, just like an insect, but for certain reasons having to do with the teeth, this is separated from the rest of insects into a group of hexapods, a separate group of hexapods, which means 6 legs. But what's neat about these guys is kind of like the pseudoscorpion, they have these pinchers, but the pinchers here at the end of the tail, what's called the circe, at the end of the diplurin. And so up here is the mouth. And the way these guys do is more like a regular scorpion, but instead of stinging, they actually reach over with the tail and they can grab prey with these circe. And it's hardened for that role. and that's why it has the pigment to it. So really, really fascinating animals. This particular one came from the lake where Dan showed the picture of the calcite rafts earlier. So there's a little continuity between these presentations. Okay. Another cave millipede. This is also a species that's known from Grand Caverns area, Madison Saltpeter Cave area. Okay. This is another type of diplurin, another hexapod. This is what's called a lidocampid. This is by a photo by a guy who is the curator at the San Antonio Zoo, this guy Dante Fenolio. And if you want to really spend some time looking at some awesome pictures, just Google this guy's name. He has pictures of salamanders and reef animals and a little book called Life in the Dark and that he put together. And Dante is an amazing photographer and promoter of biodiversity. So if you get a chance, go look up Dante's stuff. But you can see, you can pretty much count the hairs on this thing, right? That's how good a photo it is. And it really illustrates troglodytic adaptations. This animal's about 1 inch long. And with these diplurans, it's hard to tell if they're coming or going when you see them because the circi actually mimic the antennae on these type of diplurans. These are among the fastest animals you'll see in a cave on a mud bank. For their size, they move almost as fast as a cheetah. They just haul across the mud bank. And they're ambush predators, and they eat things like springtails and other smaller cave-adapted animals. All right, this is another picture of a geopidgid. It's better than the one I had, but this one's not from Virginia, so I wanted to show you my Virginia example. But again, it's got these circi here, kind of like you'd see on an earwig. But in geopidgid diplurans, these are used for predation. All right, so now let's go underwater. Let's see what's living in the water. So the most common thing we're going to see are crustaceans. We're going to see amphipods, which are side swimmers, also called scuds. You'll see them on the surface. You see them at the beach. Some of the things we call sand fleas are actually amphipod crustaceans, and some of them are amphibious. They'll be in and out. The cave ones are aquatic obligate in general. There's one in the Shenandoah Valley you'll see sometimes crawling from pool to pool as it hunts its smaller cousins. So pretty diverse. We got 36 species in the Virginias. Again, there's a bunch of undescribed species as well. So once you add them up, say at least 50 species of cave amphipod. The isopods, a little bit less diverse at the species level, but there's more diversity at the genus level. We've got Lursius I showed earlier. This is a genus called Sachydodia, and we'll see the Madison Cave isopods genus in a little while called Antrolena. There's also smaller crustaceans called copepods that you'll see. And they're, again, this is 0.5 millimeter, so they're tinier. If you ever did like the life in a pond thing when you were a kid, you would see the one called Cyclops. Well, Cyclops is a copepod. And you'll see lots of these. I pulled a jar of water out of a well over at Cremora, just a little south of you guys on the edge of the Blue Ridge, about 10 years ago now. And it had 75 of these guys in it. So these guys are around, and they're in your aquifer, they're in your water. And, so when you drink that well water there in the Shenandoah Valley, you're probably definitely drinking these guys. And sometimes you might see one of these guys, or you might end up swallowing it before you see it. But we'll occasionally get calls of these animals showing up in people's wells. It was more common when more people had hand-dug wells or driven wells. The modern drilled well with a screen it only brings the smaller stuff in. Okay, and I mentioned gastropods. I apologize for the lousy photo. I'll get you some better ones next time. But this is a cave snail. There's a couple dozen cave snail species in the Appalachians, and these are really interesting. Snails are generally considered to be sensitive receptors, particularly native snails. So they provide a barometer or a measuring point for water quality. If the snails go, you probably have something going on with the quality of your water. So going back to the amphipods, again, side swimmers or scuds. Most of the cave adapted ones are the genus Stygobromus. This is one looking up out of that pool from beneath the calcite raft that Dan showed earlier. And actually, that's a photograph in the field, which are hard to take. Again, we got a bunch of different species, and they're sensitive receptors as well to groundwater contamination. Here's a really good picture of one though, so you can get an idea what these things actually look like. If you put this up to the size of a great white shark and had it do battle, it would destroy the shark. These things are amazing animals. They've got all kinds of sharp bits and pokey bits, and they are predators. They eat flatworms, they'll eat smaller amphipods, they eat copepods. Pretty interesting animals. They also actually have a diverse of ecological roles. I say they're predators, they're not all predators. In the Edwards Aquifer alone in Texas, there are 14 different species of groundwater amphipod, and all 14 occupies its own unique trophic level in that system, meaning what they eat. So there's a whole world down there that we just know a little bit about. This is a close-up of an isopod of the genus Cicadodia. These guys, while amphipods, these guys are free swimmers. These guys aren't. These guys are benthic creatures. They crawl around the bottom, and if they fall off the wall, they'll just kind of glide down, and they'll start crawling again whenever they land. They're not strong swimmers at all. But these are the most common isopod that you'd see in an Appalachian cave, and they're very common in Shenandoah Valley caves. Okay, this is Lursius. This guy is only known from the upper Tennessee River basin. So that would be south of the New River drainage in caves. In the springs up there in the valley, you'll see the cousin of this guy, a little isopod called Lursius brachyurus, but they're not known to inhabit caves in the Shenandoah Valley. In the upper Tennessee River basin, however, there's several cave species and a bunch of different spring species. It's a big mystery why they're so different and more diverse in that area. This is the poster child, though, for cave fauna in Virginia. This is the Madison Cave isopod. This is the one that all the consultants know about, because whenever they have a project that has federal money attached to it, they get a little nasty gram from the Fish and Wildlife Service that says, you need to protect the Madison Cave isopod. And then they call us up and they say, well, what can we do to protect the Madison Cave isopod? And we're like, well, don't fill sinkholes, don't pump too much groundwater, don't build stormwater basins that aren't lined so that are going to collapse, and stay away from certain types of sites. So we try to provide some benefits. A lot of the things that impact this animal aren't really things that are regulated, and I'll mention more about that later. But what's so special about this animal is that it's the only representative of its genus. Like, I was talking earlier, you got 53 different pseudoscorpions and a couple dozen Cicadodia and blah, blah, blah. Well, this thing, when it was discovered in 1958 at Madison Saltpeter Cave, which is a cave right by Grand Caverns, when they discovered it, they didn't believe their eyes, because this was unlike anything that they had seen in the Appalachian cave fauna. It was free swimming, and the closest place anybody had ever seen anything like this was in Texas. Okay? And everywhere else in the world where you have isopods that look like this, they're called cirrholandid isopods. They're very close to the modern ocean. They're very close. They had a recent close connection to the ocean, because where you really see these guys is on coral reefs. These animals are extensive reef predators. If you've got a saltwater fish tank and you get an infestation of this guy's cousins, you pretty much have to start over again. They're just they're kind of a pretty bad parasite. If you're trying to keep pet fish. As far as the role in the ocean is, they're an integral component of the reef ecology. But in places where limestone and karst areas have come in contact with seawater, these ancestors have invaded, the ancestral species have invaded these karst aquifers. And so the implication of this for the Shenandoah Valley is that long, long ago, the ancestors of the Madison Cave isopod invaded from ocean water that was in contact with the Shenandoah Valley. And I think that's Occam's razor. I think that's the most reasonable explanation. Some of Dan's colleagues at the USGS have proposed that the Chesapeake Bay impact crater created a tidal wave that pushed the isopod over the Blue Ridge. I thought they weren't serious. I hope they're not serious. But, maybe I'm just not a creative thinker. I think this just reflects that we did have ocean water in contact with the Shenandoah Valley at some point, tens of millions of years ago. So this guy really is a living fossil. And there'll be a little bit more about it later. All right, and so here's another nice picture in the field of some isopods. These are the ocellid isopods, or the more common types. And here you see both the Sacerdotia and the Lursius together on the same rock. You don't see that too much. That's in a cave in Lee County, Virginia. Okay. So now we're going to talk about a little bit about conservation science. So in my agency, and with the Nature Conservancy, which is sort of the private nonprofit version of what we do at Natural Heritage, we talk about conservation status. And conservation status is mostly related to rarity. So if you look here, you can see G1 is something that's considered to be critically imperiled just because of its rarity. So if you have 5 or fewer populations of an animal globally, and some other factors like steep declines, disease, etc., that can get something this critically imperiled status. As you'll see in a minute, a lot of our cave animals have this status. G2 means it's imperiled, again, by virtue of number of populations or by threats. G3, vulnerable, but probably okay. G4, apparently secure, and G5, it's everywhere. Okay? Human beings are at G5. Although one can make the argument that maybe we're critically imperiled due to our own behaviors. But that's a philosophical argument. From a distribution perspective, we're a G5. So let's go here. Okay, so if we look at our troglodytic species and we throw them all in cans that are labeled G1 through G5, this is what we see. We see that 81 of them go in the can labeled G1, mean known from 5 or fewer locations. Our karst fauna, by the standard methodology, is a very endangered fauna. 118 of those species are a G1 or G2. Yeah, it's, over half of the species, about 2/3 of the species we know are considered imperiled or critically imperiled under the standard conservation methodology. And again, this has to do with this high degree of endemism. The stuff you find at Shenandoah Caverns is different from the stuff you find at Endless Caverns. They're different animals in the two things, related but different species. And that's because those caves are isolated from each other. Okay, so how do we protect this stuff? Well, there's 2 ways to protect animals. One is you protect them based on the species. It's like, if I've got this species here, I'm going to protect its habitat. And the 2 big hammers that the legislatures and all their wisdom have given people to work with are the Endangered Species Acts and state wildlife laws. So the Federal Endangered Species Act, and there's a state one as well, basically, it's focused on species level protection. We're not going to do anything special for the habitat unless something rare is there, and unless we've decided not only is it rare, but it's worthy of designation. That's one category. We'll park that on the side for now. The other category is habitat-based protection. In other words, these habitats are worthy protecting because they have a diversity of things in them, and we don't care whether they're rare or not. We just want to protect the habitat because it's a valuable habitat. And this is more like general environmental laws, your TMDLs, impaired streams, that kind of stuff. So this is our 2 ways that we really get protection of rare animals or threatened, imperiled animals. So the problem with the first one, the Endangered Species Act, is there's almost none of those species on the Endangered Species Act. We've got 2 federally listed cave obligate species out of that 168 in Virginia, and we have a handful of state-listed species, which is a much lower level of protection. So really, very few species protections are afforded. The 2 that get the species protections are 2 you've seen already: the Lee County cave isopod, which exists just in a very small area down in Lee County, called Lursius usdagalin. And then we have Antrolena lyra, which is the Shenandoah Valley endemic cereal-landed isopod. Now, I will tell you, I hate to disappoint you, but in the New Market area, you're very unlikely to pull one of these out of your well. Okay? Keep looking. If you find one, I'll buy you a pizza. It'll be great, because I'd love to have one from New Market, but we've sampled a lot in that area, and we've never found this guy. And I think it's the North Fork Shenandoah watershed, for some reason, had an event that resulted in these guys no longer being there. And I think that was before humans ever got here. I think it was a geological event. But anyway, we can pontificate about that at a later time. The thing is, these are two species. They're both isopods. They're both cave isopods. They're both in the same region, and they're completely different animals when it comes to trying to protect them. The one at the top, very small area, you literally can afford to buy the entire range of the species. And the state owns, between the state and the Nature Conservancy, we own every cave or spring this thing's known from now. Okay, so this guy, you can really focus and just own it and protect it. Madison Cave isopod exists, now we know it exists from Lexington, Virginia to Charlestown, West Virginia. It's really hard to find. This guy, if you have a cave and you have a flowing stream, he's there if you're in his range. This guy, you might have to sample 5, 10 times before one of them comes swimming by and jumps in your trap. So that's just two of the differences between these two that make your protection strategies very different. So here you can see Lee County isopod. There's the global range, that little green blob I hope you can see down the lower left-hand corner, tiny. This is where the Madison Cave isopod is known from here, and it extends into West Virginia. I was too lazy to make a better range map yet. But again, you can see, and of course you're in Newmarket, you're kind of right here where I wouldn't be shocked if you found it, but I probably would actually be kind of shocked. This is sort of the range where we know it's present. Okay, so the characteristics in common between these two species is they're cave animals, they're endemics, they're rare, and they're federally protected. But we'll look at the differences briefly just to illustrate that this stuff can be complex. Again, this guy's widely distributed in the Shenandoah Valley. They get to be, typically they're a centimeter long. The longest one on record is 21 millimeters, so it actually can get as much as 2 centimeters long. And it's the only one of its kind. You know, to get to the closest related thing, you got to go to Texas. And DNA evidence suggests that those two species lines split 28 million years ago. So if you think of like DNA.com, where you and somebody in Iowa share an ancestor from Dublin from 50 years ago or 150 years ago. Okay, if this guy and the Sierra Leoneans in Texas share an ancestor from south of the equator 28 million years ago. So their ancestry.com is a lot more distant than it is for us and our relatives from Europe or whatever. But it's the same technology we use to do those analyses. It's the DNA. Okay, one of the fun things about having a federally listed species is you get goofy things like this. This is a power plant up in Warren County that Dominion has. It's Dominion Warren's gas turbine-fired power plant. It was put in about 10 years ago. And they actually built this right by a Madison Cave isopod cave. And because of that, we got all kinds of extra precautions taken to how they handle their stormwater. They also do a lot of monitoring. But one of the funny things is everybody who sets foot on Dominion Warren has to go through Madison Cave isopod training. And the first thing you see when you walk in is a picture of the Madison Cave isopod, but it's anatomically incorrect because it has eyes and it also has Popeye forearms. But outside of that, go Dominion, try to protect the isopod. But there's all these, you have to literally sit through a 20-minute video and know what to do if you were to encounter an isopod on the power plant site, which is extremely unlikely because they're 100 feet below the site and they're also off to the east. But anyway, it's kind of entertaining. So this Madison Cave isopod, the way you find it is you drop baited traps in most places. This is a place called Stager's Fissure up near Grand Caverns. You can see here that jar, which Dr. John Holsinger is holding, is full of isopods. So places where you find these things, they can be locally abundant. That particular place is sort of a natural baited site because debris comes down the side of the hill there into the cave at the base of the hill by the South River. And then all kinds of things fall in there. These isopods are just very, very well fed at this area. Turning over to the Lee County Cave isopod, completely different ball game. Again, it's instead of being in deep water where you have to drop traps in, I found the Madison Cave isopod over 100 feet deep in people's wells under the water surface in still water. This guy you only find in fast-moving shallow water. It can't live in pools. There's not enough oxygen and there's too many predators. And this guy was listed as endangered because one of the two caves it was known from at the time got contaminated by leachate from a sawmill. And the leachate basically took all the oxygen out of the water, which killed off that population. And you can see here, you'll actually find this just on damp surfaces sometimes. It's aquatic, but it's almost to the level of being amphibious. When this dries out, they'll just encrust themselves. And then when it gets wet again, they start crawling around. Discovered that one day when we thought a bunch of them had died until we threw a little water on them and they started moving. It was very scientific. And so with the species-based protection, again, 8 out of 167 species have species protection. Only 2 of them are federally listed. That's where all the money is. And what it's done to us in a conservation biology way is it's really skewed where our conservation efforts are. You work where the light is, you work where the money is. And so because we have money to study these areas where these species are, we keep going back over and over again. And this handful of species gets a lot of protection while the vast majority of the species are ignored. And that's a real problem if you consider that earlier methodology to have some validity, which I think it does. So let's go back now and let's look at this biogeography at a broader scale. I've mentioned about protecting them. Let's just talk about how diverse it is at the scale of the Appalachians. So this area, we could argue about what the Appalachians is. For purposes of our discussion, the area inside the black is the Appalachians. Okay. And so in that area, what the green represents is everywhere anybody ever collected a cave-limited animal. And this just goes to show how many people there are on the earth and how many people have free time to go collect cave animals, because that's a huge number of collections, right? That's a lot of dots. Who would have thought that many cave animals had been collected over the years in the Appalachians? But that's what we've got. And you can see over here, this list of illustrious people includes Dan Doctor, whom you heard earlier. I actually had the dubious distinction of serving on the advisory committee for this project, and also like working behind the scenes to actually get them to give money to it. So it was pretty funny, me actually getting to review Dan's work. It was quite humorous. A lot of these guys here are kind of mentors to me, and I was glad to be able to give them money. I was glad to be able to give them a hard time. So if we look here, there's the terrestrial records on the right. Here are the aquatic records on the left. And what the gray is where the karst bedrock is. This is from Dan's National Karst Map, his and Dave Weary's from the USGS. And you can see there's a pretty good coverage in terms of cave animals. And what you'll see is Virginia's got a lot of records. So does West Virginia. You see the records drop off in Pennsylvania. That's because of glaciers. When the glaciers covered up through central PA and all that cold water and everything and scoured the surface off, it really did a number on the cave-limited fauna. You find most of the cave-limited fauna south of the glacial front. If we try to figure out a way to measure this, we measure the species richness, which is measured by the number of species in one of those grid cells. You really start to see where the biodiversity hotspots are that we talked about earlier. And you can see in Virginia, you've got a hotspot down, well, actually Greenbrier Valley, West Virginia. You got a terrestrial hotspot down in Lee County. But if you come over here, you can see that in the Shenandoah Valley, you start picking up some hotspots in the aquatic fauna. And it kind of makes sense. And this hotspot's going to grow bigger over time because of some increased sampling in the Shenandoah Valley. And the reason we have more aquatics in the Shenandoah Valley is we have more aquatic habitat. As we were talking about earlier, a lot of the karst in the Shenandoah Valley is below the water table. There's a lot more room for aquatics and not as much room for terrestrial stuff as there is compared to down here. Some other hotspots you'll see are down here where Tennessee, Alabama, and Georgia come together in the tag area, and that's reflected in the aquatic and terrestrial. And then out in the Mammoth Cave area in Kentucky, which bleeds over into southern Indiana, that's another area of hotspot. Sometimes these hotspots are actually driven by where people live. Dr. Jerry Lewis lives here. He does a lot of biology, so probably more stuff is found there because he looks harder. This hotspot in Southwest Virginia, part of that's probably due to the fact John Holsinger used to work at a university right here in Kingsport, Tennessee, or Johnson City, Tennessee. So these maps, you have to approach them with a cautious eye because a lot of them are not just maps of where concentrations of animals are, but also where people looked. One of the really neat things I like to do is go in a little bit closer and take a look at the relationship between the distribution of cave animals and other landscape features. And this is an example from a paper that Jerry Lewis and I have that we're putting out now. And it's just basically talking about the distribution of isopods. And again, this should not be shared. I know this is recorded, but this is not really for sharing per se, because it's not been published yet, but it's in press. So please be mindful of that. But what you can see here is these different isopod species occupy different drainages and different geologies. In the Greenbrier Valley, we have this one Cicadodia species. In the New River Valley and Roanoke Valleys, we have another one. When we come down here, west of the New River, we get into one that only exists west of the New River within the diamond-shaped one, which is what's called the cannula group. If we go down here, we see these little circles represent a different group of isopod called the Stygia group. And they're all known from just west of the New River. Scattered in here are these diamonds, or these triangles, which represent Lursius species in caves. And then you've got these crosses, which represent some sort of oddball occurrences, which in this case are a third group of isopods in the same genus as the diamonds and the circles. And the point of this is there's a systematic pattern to these distributions. And this tells us a lot about the geologic history. And that in turn sort of can inform us in terms of how things need to be managed and what's really going on in terms of the natural history. I'm not particularly good at telling you why it's of practical value. So if you want that, you need a better BSer than me. I just think it's really cool, and it tells us a lot about the natural history. And this guy in particular is fascinating to me because it's the only one that makes it into the Upper Tennessee River Basin in this whole species group. And that's telling us something profound. I'm not sure what it is yet, but I'm sure it's profound. A couple of things we don't have in Virginia in terms of cave life are cave crayfish. They're only known from the interior lowlands, and this one population that's kind of weird is cave limited, but doesn't look all that cave adapted, from out near Sharps Cave area in the northern part of the Greenbrier Valley. And then cave fish, we don't have any. We have fish in caves, but they're surface fish. The cave fish seem to be limited to, again, the interior lowland plateau where Mammoth Cave is, Middle Tennessee. What we do have that these guys don't have is that Madison Cave isopod, which is right there on your all's doorstep in the Shenandoah Valley. So when they start bragging about their cave fish and their cave crayfish, we can give them the raspberries and say, well, you don't have any serralanded isopods, and that's more interesting anyway. All right, so there's a picture of a blind cave fish. Everybody likes to see those. They're really, really cool. And there's a few different species, a couple different genuses of these. We don't have these, again, in Virginia's caves, or at least we haven't found them if we do. But they're fascinating animals. No eyes, and they actually have selectively developed hearing loss, where they've lost their hearing in the frequency range where most of the noise is in caves. So they're able to ignore the noise because they can't hear the noise. And they only hear in frequency ranges where there's less activity. It's really cool adaptations. When you study cave life, that's really the value of this biodiversity to humans, is there's all kinds of crazy genetic stuff going on that may well benefit people someday in terms of understanding biology and also in terms of pharmaceutical solutions. They also have cave-adapted salamanders. Again, we have cave salamanders, but we don't have ones like this that are adapted for life in caves. This is the Tennessee cave salamander, Gyronophilus pallucus, which is a good name for somebody from Tennessee. And anyway, last thing is, if you want to get more in tune and get a really nice book about cave life in the Virginias, the Cave Conservancy of the Virginias and the NSS worked together to put out this book called Cave Life of the Virginias. And if you go to the National Speleological Bookstore, you can Google Speleo Books or NSS Bookstore. They have these for sale. And it's a really nice book that kind of gives you a better introduction than I just did on cave life in the Appalachians. And with that, I will entertain any questions you all may have. No questions? Oh, wait, let's look in the chat. Let's see. Oh, Andy. Yes. Andy is awesome. So Andy is Stygobromus hubbardi. And Andy was named after a guy named Dave Hubbard, who was the man who first collected Andy the amphipod at Hupp's Hill Caverns. So Crystal Caverns at Hupp's Hill was talked about. It's a stop on the Karst Trail. And it's a pretty neat place. And unfortunately, right now it's not open. I think the town of Strasburg, if we're lucky, at some point we'll be able to come up with a way to allow some visitation to Crystal Caverns at Hupp's Hill. I think it would be good for the town, and it would be good for public education to be able to get in there. It's a really cool cave. It was the first cave I was ever in was Crystal Caverns at Hupp's Hill, because I was from Strasburg. But anyway, Andy's still there. Andy's alive and well. We collected some specimens of Andy about 2 years ago for genetic analysis from a little pool that's just about 50 feet inside the entrance. There's another cave there where Andy's known from. And then we found a population of somebody who looks a lot like Andy in a cave called Beans Cave in Frederick County. So Andy the amphipod still seems to be doing okay.
All right, are we have any more questions? Well, we've had some really high quality presentations this afternoon. I'd like to thank Will and Meredith and Dan for such good content and for such good partnership. And I'd also like to thank our guest today. This is, you've helped us pull off our first Zoom library program. I'd like to remind you that our next library lecture will be on July 11th, and it will be a presentation from the authors of the book Bad Moon Rising. So be watching for an email that will come out once we have edited the recordings. We will send you a link to the recordings in case you want to share this information. That email will have some additional resources in case you'd like more information. And I hope everybody's going to go find that brochure and start checking off all the caves they're visiting. So thanks again. Have a great weekend, and we'll say farewell.